Today, I am going to discuss a very important aspect of biology, that is reduction potential. Even it is the concept from core chemistry, it is essential to understand the definition, the calculations, and applications of reduction potential in biological processes. I call reduction potential as one of the divines of biological system because along with entropy, enthalpy, free energy, reduction potential is one of the parameter which is actually governing different type of biological processes, as well as there are grand designs of biology like light reaction of photosynthesis, mitochondrial oxidation phosphorylation are mainly crafted around reduction potential as a core concept. So in this session, we are going to understand the definitions, we are going to understand the applications, and we are going to understand how reduction potential is governing very complex biological processes. So let's start with the definition of reduction potential. So I use a very easy but an amazing definition of reduction potential because it never fails us whenever we want to apply reduction potential into understanding a core biological concept. The definition says that reduction potential is nothing but tendency of accepting the electrons. That means the value of reduction potential is actually defining the ability, the keenness, the interest of keeping the electrons. Imagine I am a chemical species and my reduction potential value is high. What does that mean? My value of reduction potential is high means my tendency of accepting the electrons, my tendency of keeping the electron, my keenness towards accepting the electron is very high. And if my reduction potential value is less, that means I am not that much interested in keeping the electrons. I'm not that much interested in accepting the electrons. So a particular chemical species, whether it will accept the electron or will donate the electron, totally depends upon the value of reduction potential. Higher value of reduction potential means higher tendency of accepting the electrons, and lower value of reduction potential is lower tendency of accepting the electrons. Now we can expand this definition by bringing some numbers. So let's imagine we are having a chemical species A with the value of reduction potential equal to 1.5 millivolt. Now in the same microenvironment, we are having another chemical species, that is chemical species B, with the value of reduction potential equal to 2.5 millivolts. Now what will happen? It is very easy to explain because the chemical species A is having lower tendency of accepting the electrons and chemical species B is having higher tendency of accepting the electrons. So electrons will be transferred from chemical species A to chemical species B to a lower value of reduction potential to a higher value of reduction potential, from a species which is less keen in keeping the electrons to a chemical species which is more keen in keeping the electrons. So electrons will be transferred from low reduction potential to high reduction potential. Now the definition is very simple, and we can also explain this definition in terms of oxidation and reduction, because if a species is losing the electron, then the species is getting oxidized or the process of oxidation is occurring. Or if the species is accepting the electron, gaining the electron, then that species is getting reduced or the process of reduction is occurring. Now one more time, the statement which is defining the reduction potential is reduction potential is nothing but tendency of accepting the electrons, or you can say the reduction potential is nothing but tendency of getting reduced. Of course, these two are same things. Tendency of accepting the electron means tendency of getting reduced, because acceptance of electrons or gaining of electrons is called as reduction. So we have learnt the definition, tendency of accepting the electron, and we have learnt another fact, that is electrons will be always transferred from low reduction potential to high reduction potential. Now let's apply this definition to understand one or two biological examples, then we will expand further and we'll do some calculations also. Now let's apply. This is a famous process in biological reactions or biochemical reactions, where NAD plus, after accepting 2 electrons, convert into NADH, and this process, or you can say the reduction potential of NAD plus in accepting 2 electrons, is equal to minus 0.32 volt. Now since there is the sign of negative, generally we think that the values which are negative will be having or will be considered as lower values, or values which are having positive sign will be considered as higher values, but that is not true, because the word higher or lower more or less are actually relative terms. To actually understand whether this minus 0.32 volt reduction potential of NAD plus is high or low, we have to bring another species and then we have to compare. So we are bringing another process in the same microenvironment, where acetoacetate is accepting 2 electrons, converting into beta hydroxybutyrate with reduction potential minus 0.35 volt. So in other words, you can say that tendency of accepting 2 electrons by acetoacetate is governed by reduction potential value minus 0.35 volt. Now if I am bringing these two processes together in the same microenvironment, what will happen? It is very easy to tell, because the reduction potential is minus 0.32 volt for NAD plus and the reduction potential is minus 0.35 volt for acetoacetate. Who is having higher value? Of course NAD plus. Who is having lower value? Of course acetoacetate. So in this whole process, if we combine the reaction will look like this. Since the reduction potential of NAD plus is high, then it would like to accept the electron. So NAD plus is accepting the electron. Since this reaction will not occur into forward direction, it will start occurring into backward direction, because reduction potential value is low. So acetoacetate cannot accept the electron. Instead of that, beta hydroxybutyrate will give the electron, because the reduction potential value is low or lower than this process. So NAD plus will accept 2 electrons given by beta hydroxybutyrate, and NAD plus will convert into NADH, and beta hydroxybutyrate will convert into acetoacetate. So the reaction direction is totally dependent upon the values of reduction potential. Here, this is the process having high reduction potential, this is the process which is having low reduction potential, and we know that electrons will always be transferred from low reduction potential to high reduction potential. So when we are combining from this process start running backwards, releasing 2 electrons. These 2 electrons are given to NAD plus, and from NAD plus we are generating NADH. Now let's see another example. We are again bringing NAD plus plus H plus plus 2 electrons with the reduction potential value equal to minus 0.32 volt. Now we are bringing another reaction into the same microenvironment, where half O2 plus 2H plus, after accepting 2 electrons, converting into water, and the reduction potential value is equal to 0.82. It is very high according to biological standards, because this reduction potential is one of the highest values in natural system or biological system or cellular system. So now what will happen? When I will combine these two reactions, this will be the final outcome. Now this process is having higher reduction potential and this process is having lower reduction potential. So electrons will not be gained by NAD plus, because it is having lower reduction potential into the same microenvironment. Instead of that, NADH will give its electron to half O2 NADH will convert into NAD plus and half O2 will convert into H2O. So for same NAD plus, we are having two situations. In first example, the reduction potential for NAD plus is higher than the other partner, and in the second example, the reduction potential for NAD plus is lower than the other partner reaction. So the lower, higher, more, less, all these words are relative. Now we already know that how to decide the transfer of electron or how to decide the directions of electrons just by keeping two things in our mind, that reduction potential is nothing but tendency of accepting the electrons, and second, electrons will always be transported or transferred from low reduction potential to high reduction potential. Now we will do some calculations where we can convert the reduction potential into energy equivalents also. But before doing that, we will be understanding a very interesting story which is happening nearly every day in our skeletal muscles as well as in the bacteria which are doing fermentation. So let's start the story and let's talk to muscles and lactic acid bacteria. So normally in muscles, the glucose is being converted into pyruvate through the metabolic pathway glycolysis, which is actually an oxidation reaction, because if you look at the chemical structure of glucose, it is an aldehyde containing alls and pyruvate is an acid. So the glycolysis is essentially an oxidation process where glucose is converting into pyruvate. Now we know from start that oxidation cannot occur alone. We have to reduce something. The system has to reduce something. So the system during this process is reducing NAD plus, which is accepting 2 electrons and converting into NADH. So oxidation, reduction, the redox pair is completed. Now the NADH which is forming into cytoplasm is going into mitochondria, losing its 2 electrons to oxidative phosphorylation, converting back into NAD plus, coming back to cytoplasm and taking part in glycolysis again. So this mitochondria is kind of recycling plant for NAD plus, which is using NADH and releasing NAD plus again in the cytoplasm. Now when we are doing vigorous exercise or heavy exercise, then after some time muscles will start facing the situation of hypoxia. Now during hypoxia, the mitochondrial component is not going to work. The reason is very simple, because the mitochondrial component is utilizing oxygen to run the oxidative phosphorylation. And since in the microenvironment oxygen is not enough, the mitochondrial component will not work. So what will happen? NAD plus, which are present in the cells, will convert into NADH, but their supply will be stopped because this recycling center is not working. Now if the supply is stopped, system has to prevent the slowdown of glycolysis. System has to run the glycolysis smoothly because exercise is going on and energy is continuously required. So system has found or evolved a very interesting method where they will reduce this pyruvate again. So the pyruvate structure is containing one carbonyl group, and they are reducing the pyruvate into lactate, the lactic acid, which is actually having alcohol, so carbonyl to alcohol. So reduction is happening. Since reduction cannot happen alone, the system has to oxidize something. So system is utilizing this NADH and converting into NAD plus by removing 2 electrons. So again a redox potential, or again a redox couple is completed. So glucose to pyruvate, oxidation, NAD plus to NADH, reduction, mitochondrial component, recycling of NAD plus, and smooth going of glycolysis. In absence of oxygen, no mitochondrial component. So we are utilizing another recycling plant which is using the reduction of pyruvate into lactate. So NADH will again oxidize into NAD plus, and this NAD plus will go back into glycolysis and will take part in glycolysis again, and the metabolic pathway will be continuously going on. So if somebody asks why muscles are making lactic acid to produce the energy, no. To run the glycolysis in absence of oxygen or in absence of mitochondrial component, where they are reducing the pyruvate into lactate, simultaneously oxidizing NADH to NAD+. It is happening in all the anaerobic bacterial system and also in lactic acid bacteria. So the lactic acid bacteria or the muscles are not producing lactate or lactic acid to make the energy. They are doing these reactions. They are doing this process to recycle the NAD plus, so their glycolysis can be continuously going on, right? Similarly, there are other bacterial strains which are doing reduction reaction in terms of fermentation. So fermentation is an essential process for the bacterial system where they have to recycle the NAD plus because they are not having a component which can convert NADH into NAD plus in presence of oxygen, right? Now, let's revise what we have learned till now. Number one, reduction potential is nothing but tendency to accept the electrons. Reduction potential is nothing but tendency to get reduced. Reduction potential is deciding the flow of electron, transport of electron, transfer of electron. And this transfer or this flow of electron is always happening from low reduction potential to high reduction potential if there is voltage difference or if there is reduction potential value difference. And this whole process is actually occurring spontaneously. So we are smelling something spontaneous. Again, we are having two species, chemical species A with low reduction potential 1.5 millivolt, chemical species B with high reduction potential 2.5 millivolt. Electrons will start moving from low reduction potential to high reduction potential spontaneously. The process is spontaneous. Whenever the word spontaneous comes into the picture of biochemical sciences, then it is always associated with release of energy. Every spontaneous process is having negative delta G, or some amount of energy will be released. Can we calculate this energy? The answer is yes. We can calculate the energy by utilizing this formula where delta G is equal to minus NF delta E. What is delta G? Free energy change or the amount of energy released. What is N? Number of electrons transferred. What is F? Faraday's constant with the value 96485 joules per mole per volt. And finally, what is delta E? Delta E is difference in reduction potential value, means acceptor minus donor, 2.5 minus 1.5. If we are utilizing this formula, then we can calculate the exact amount of energy released during the electron transfer from a species which is having low reduction potential to a species which is having high reduction potential. So this is also one of the method utilized by our cellular system to make the energy. So whenever the energy is required, what cells are doing? They are taking two species or more than two species. They are making a chain where the electrons are moving from low reduction potential species to higher one, then to next higher one, then to next higher one. So when these electrons are moving, energy is being released, and this released energy can be utilized to do different other types of biochemical reactions or other types of biochemical processes. Let's calculate the amount of energy for a famous example which we have already seen. So we have seen that NAD plus H plus 2 electrons reduction potential minus 0.32 volt, and half O2 H2O reduction potential is plus 0.82. The final reaction will happen where electrons will go from NADH to half O2, NADH converting into NAD plus, and half O2 is converting in water. The famous reaction of mitochondria. In mitochondria, the electron transport chain start from NADH. Electron transport system start from NADH and ends at H2O. The NADH is giving the electrons. Electrons are passing through different transporters and then finally reaching to oxygen, which is the final acceptor of electron. How much amount of energy is released? Can we calculate? The answer is yes. Delta G is equal to minus NF delta E. Delta G is equal to minus 2. 2 electrons are being transferred. F is Faraday constant, that is 96485. What is delta E? Acceptor minus donor, 0.82 minus 0.32. The answer is minus 220 kilojoule per mole. So the amount of energy which is released in electron transfer from NAD plus to oxygen is equivalent to minus 220 kilojoule per mole. So again, tendency of accepting the electrons, tendency of getting reduced, electrons are going to be transferred from low reduction potential to high reduction potential. We can calculate the energy by using the formula delta G is equal to minus NF delta E. Till now, whatever we have learned is restricted to 1 or 2 step processes, which are simple to understand. But there are some grand biological designs built upon the concept of reduction potential, and now we are going to discuss them. So first design is the amazing light reaction of photosynthesis or Z scheme, and the second design is oxidative phosphorylation. So we'll start with how the Z scheme or light reaction is crafted by utilizing reduction potential as a central concept. So let's understand first what is interesting about reaction of photosynthesis. If I look at the reaction of photosynthesis, then it's a nice reaction which is giving us one carbohydrate, one glucose molecule by using 6 CO2. Now, can I dissect this definition using reduction and oxidation? Answer is yes. So I can calculate the oxidation number on carbon in CO2 molecule. So carbon is X, then 2 oxygen atoms, 1 oxygen is minus 2, so X minus 4 is equal to 0. I can also calculate the oxidation number on carbon in glucose molecule. 6 carbon, that is 6X, then 12 hydrogen, that is plus 12, 6 oxygen, that is minus 12, equal to 0, X equal to 0. So logically, photosynthesis is a process where the oxidation number of carbon is changing from plus 4 to 0. Then how do we reach from plus 4 to 0? The answer is simple, by giving 4 electrons. So positive 4, negative 4, resultant is 0. Now since there are 6 carbons which are undergoing this process, so we will be requiring 24 electrons. Now I can define the photosynthetic reaction as a reduction reaction where 6 CO2 or 6 carbons of 6 CO2 molecules are getting reduced into one glucose molecule by accepting how many electrons? 24 electrons, right? Gain of electron or acceptance of electron is nothing but reduction. Now since these electrons are being utilized, 24 electrons are being utilized, they should be coming from somewhere, and Mother Nature and evolutionary forces at some time point have decided that water should be giving these 24 electrons, because they thought they can take 12 water molecules which can give us 24H plus, of course, 6 CO2 which are being released here, and that will give us 24 electrons. So I can again define the photosynthesis by saying that photosynthesis is a reduction reaction where 6 carbons of CO2 molecules are getting reduced into one glucose molecule after accepting 24 electrons which are given by 12 water molecules. So the whole dependency of doing photosynthesis is on water molecule. That 12 water molecule will come and will give 24 electrons, and we will run this reduction reaction, and ultimately will create glucose for our life. Now they have chosen a wrong thing actually, or a difficult thing to do. Why? Because we have already seen that this process where half of O2 is accepting 2 electron and converting into water molecule is having the highest reduction potential, that is plus 0.82 volt. It means it is impossible task to take out electrons from water, because it is having very high value of reduction potential, and reduction potential is nothing but tendency to accept the electrons, right? So they are very keen in accepting the electron. They will never be interested in giving up their electrons, and we are making our life dependent upon the fact that 12 water molecules are going to give us 24 electrons. Very interesting things going on here. So they have decided that we will create an amazing machinery, an amazing design by which they will be able to take out these 24 electrons. But interesting things are not stopping here. It gets interesting further. So the whole photosynthesis is divided into two parts. From 12 water molecules, 24 electrons have to be given to 6 CO2 molecules. And this electron transfer is not direct. First, the 24 electrons are given to 12 NADP molecules, NADP plus molecules, which will convert into 12 NADPH, that is step 1. Then this 12 NADPH will convert back into 12 NADP plus, will release 24 electrons, and those 24 electrons will be given to 6 CO2. So we are having a pit stop here. We are having a central point of the electron transfer in photosynthesis, where the NADP-NADPH couple is behaving as a mediator of electron transfer. First 24 electrons, NADP plus to NADPH, then reversible reaction, then those 24 electrons will be given to 6 CO2. So ultimately, 24 electrons are being transferred from 12 water molecule to 6 CO2 in a two-step manner. And the first step is dependent upon light, so we are calling it as light reaction. And the second step is not depending upon light, so we are calling it as what? Dark reaction, right? Now, what is interesting here? We already know that it will not give you 24 electrons because reduction potential is very high, and the reduction potential of 12 NADP plus to NADPH is minus 0.32. So the light reaction is actually giving 24 electrons from high reduction potential to low reduction potential, or you can say a highest value of reduction potential to a very low value of reduction potential. So are we going against the law of physics and law of chemistry or law of reduction potential when we are doing light reaction? The answer is no. Now comes one of the best design, one of the mesmerizing designs of biology, where 24 electrons will be taken out from water without breaking any physical or chemical law, and that design is called as Z scheme. And whenever I learn it, whenever I try to explain it, I always get the goosebumps that how to accommodate the principle of reduction potential. The systems are using many proteins, many electron acceptors, donors, chemical molecules, just to do one thing, that we have to take out 24 electron without opposing any basic fundamental law. Now let's see how the design works. And to understand the design, we are going to utilize a graph, and by using this graph, we are actually praising the evolution and Mother Nature. So what is that graph? On x-axis of that graph, we are having reduction potential. The central value is 0 volt, then minus 1.6, and then plus 1.2. So when I am going upwards, the reduction potential is decreasing, or when I am coming downwards, the reduction potential is increasing. Now we are having water here, that is, water is giving us 2 electrons, and the reduction potential of this process is plus 0.82 volt, which we have already seen. So the position of water on this graph is here, right? Now we have to give these electrons to NADP, which is having reduction potential of minus 0.32 volt. So the position of NADP plus is here. Now the electrons have to go from here to here, from high reduction potential to low reduction potential, which is not possible without making any exception. But we are not going to do that. We are going to understand the design created by Mother Nature to analyze why these electrons are able to go from high reduction potential to low reduction potential. So the whole story is starting from a chlorophyll molecule, the reaction center of photosystem 2, so-called photosystem 2. We are calling it as P680. Now you can see that the position of P680 is here on this graph. It is having lower reduction potential than water, because when we are going down, reduction potential is increasing. So electrons cannot go from water to P680. So in step 1, P680 will absorb a photon and will change its position on the graph here. So changing the position means actually after accepting the photon, after absorbing the photon, the reduction potential of P680 is decreasing, because when we are going up, reduction potential is decreasing in the graph. In cell, the position of molecule is not changing. When I am moving this molecule here, that means I am not changing the position of molecule inside the cell. I am changing the value of reduction potential for that particular molecule, or you can say the position is changing on the graph, not in a biochemical pathway. Now the P680 is called as P60 star, 680 star, that is excited P680 with lower reduction potential. Nearby a molecule is present which is called as pheophytin. Now if I look at the position of pheophytin on this graph, I can see that pheophytin is having higher reduction potential than P680. Why high? Because when I go down, reduction potential is increasing. So there will be automatic transfer of electron from P680 to pheophytin. Once the P680 is losing its electron to pheophytin, again the position of P680 is changing. Now it is reaching here downwards, and an amazing transient chemical species is generated, which is called as radical ion, which is having its single electron and also having positive charge because it has lost one electron. And this P680 radical ion is having reduction potential equal to plus 1.2, which is one of the highest in natural system. So what will happen? Now water has found a species nearby which is having higher reduction potential, so it has to give its electron. Now this P680 will accept the electron from water, because now the electron can be given from low reduction potential to high reduction potential, and it will convert back into P680, which is normal. The cycle is ready to go again. Absorb the photon, become excited, lose the electron, become radical ion, have highest value of reduction potential, and snatch the electron from water. You can do it continuously, 24 hours, every second, whenever photons are present. Now, some people can ask, before going ahead, I will be giving explanation of one minute thing. Some people can ask that why absorbing the photon is reducing the reduction potential of a molecule. To understand that, we can go back into our older classes, and there we have seen that whenever the electrons are absorbing the photon, they are moving from outer shell from they are moving to outer shell from inner shells. So when the electrons are present in inner shell, they are near to nucleus. The bonding or the binding force of nucleus or the attraction force of nucleus on electron is high. So it is difficult to take them out. Difficulty in taking the electron out means, of course, the reduction potential is high. The atom is not interested in giving the electron. But after absorbing the photon, they are becoming excited. Excited means electron is moving to outer shells, moving away from the nucleus. Moving away from the nucleus means attraction force is less. Attraction force is less means now the electron can be easily plugged out. Easy removal of electron means reduction potential is low. So they are changing the position because electrons are absorbing the photon and moving between the energy shells. Now let's continue. Near phosphatine, another electron acceptor is present, then another, then another. Ultimately, it is reaching to plastocyanine. You can see that the position of plastocyanine explains that plastocyanine is having higher reduction potential than phosphatine. So ultimately, electron is going to reach to plastocyanine. Now again, we are stuck in the same problem. Electron is here at higher reduction potential, but it has to go here, which is having lower reduction potential. So how the electron should be transported from plastocyanine to NADP plus, how it should be transported from high reduction potential to lower reduction potential. So we'll bring another reaction center, P700. What will it do? Accept the photon. After that, excitation, P700 star. After that, electron can be given to iron sulfur complex, a high reduction potential species, and from iron sulfur complex it will reach to NADP plus. But after losing the electron P700, the P700 will convert into a radical ion, P700 dot positive charge. It is having higher reduction potential than plastocyanine, so it can snatch the electron from plastocyanine, can convert back into P700 and ready to do next cycle. 2 times. Everything is happening 2 times. And from this iron sulfur complex, electron will be given to NADP plus. NADP plus will convert into NADPH. Amazing design. Molecules are changing their positions on the graph. Molecules are changing, are so dynamic in terms of their reduction potential to do only one thing. Snatch the electron from water molecule and give those electrons to NADP plus. To do an uphill task where electrons are flowing from high reduction potential to low reduction potential by utilizing light, by utilizing chlorophyll. So somebody asked, what is the role of light and chlorophyll molecule? Then the answer should be in the terms of reduction potential. The answer should be they have to generate a machinery which is having molecules with the value of reduction potential much higher than water. So ultimately the water will start giving electrons to NADP plus, or you can say the electrons can be transported from high reduction potential to low reduction potential. This design is looking like a Z. So we call it as what? Z scheme. The Z scheme is not a biochemical pathway. It's a graph where the molecules are having dynamic values of reduction potential and electrons are being transported from water to NADP+. Now there is a number problem and we have to understand that also how we are going to solve that particular number problem. Now the Z scheme is looking nice, but there is a problem and I call this problem as the problem of numbers. You can see that the whole Z scheme, the whole system is able to transfer one electron at a time. But to generate one oxygen molecule, two water molecules has to be utilized and simultaneously 4 electrons will be released. So how to balance this number? When the pathway or the system is transporting 1 electron at a time, but the donor is giving 4 electron at a time. So let's solve this number problem. So the whole thing is happening into enzymes called as photosystems. Now you can see that P680 is absorbing the photon, converting into P680 star, losing electron to pheophytin, coming back to P680 radical ion or P680 dot positive. Now this dot positive is not accepting the electron directly from water. It is accepting the electron from a tyrosine residue called as tyrosine Z, TYRZ, of the protein system and converting back into P680. So the one electron which is gained by P680 radical ion into the Z scheme during the Z scheme is not coming directly from water. It is coming from tyrosine Z residue. After giving this electron, tyrosine Z is becoming the tyrosine Z radical, and this radical is not taking electron from water but taking electron from a complex of manganese 4 calcium. So there are 4 manganese. One can give the electron to tyrosine Z radical, which is converting back into tyrosine Z. Now second time the second electron will come from manganese, third time the third electron will come, fourth time the fourth electron will come. Ultimately, once the fourth electrons are donated by, or 4 electrons are donated by manganese 4 calcium complex to the Z scheme, then the whole complex will accept 4 electrons which are simultaneously released by water. So this is how the number problem is being solved, and we call this whole thing as oxygen evolving complex, which are having intermediates between water and P680, where 4 electrons can be simultaneously transferred to manganese 4 Ca, which is giving 1 electron at a time to tyrosine, and tyrosine is giving 1 electron at a time to P680. So this was the beautiful design of Z scheme, which is governed by the principle of reduction potential. And the aim of this design was how to transport the electron from water to NADP, or how to transfer the electron from high reduction potential to low reduction potential. The second design is the design of mitochondrial oxidative phosphorylation. But before that, I will again assert that this scheme, Z scheme, is actually a graph, and it looks like this in the biological system. So in pathway, the molecules are not changing their position. They are changing their position according to reduction potential value in the graph itself. So there is a difference between the graph and pathway. Now let's see the second grand design, that is mitochondrial electron transport, and which is easier than the photosynthesis because we will see what is happening here. So the mitochondrial oxidative phosphorylation is occurring when NADH is giving its 2 electrons to oxygen, and oxygen is converting into water, and amount of energy which is being released here is minus 220 kilojoule per mole. Now system doesn't want to release this much amount of energy simultaneously because that can damage the tissue, it can damage the cells, it can generate the heat. So system wants to create the energy in small packets. So the whole process is starting from NADH and ending at O2, which is converting into H2O after accepting the electrons. So from NADH, 2 electrons are transferred to a protein complex, electron transport complex 1, which is present into the inner mitochondrial membrane. The reduction potential value of complex 1 is higher than NADH, so of course the transport of electron is spontaneous. The second complex, the third complex, the fourth complex are arranged into the inner mitochondrial membrane. So from NADH, 2 electrons are going to complex 1. Then from complex 1, 2 electrons are going to complex 3. From complex 3, 2 electrons are going to complex 4, and finally they are going to oxygen. So NADH 1, 3, 4 oxygen, they are arranged in a system which is following the increasing order for reduction potential values. So they can automatically be transferred. NADH to first, automatic transfer, first to third, automatic transfer, third to fourth, automatic transfer, and fourth to oxygen, again the automatic transfer. So in every step, a small energy is getting released, which is utilized to transport the protons from mitochondrial matrix to intermembrane space, which we will see in our upcoming lectures when I will be discussing the complete mitochondria oxidative phosphorylation, ETS, and everything. But every time 2 electrons are being transferred, energy is released, and that energy is utilized to transport the protons from mitochondrial matrix to intermembrane space, and these protons will be coming back again and will be generating ATP. That's the discussion for another day. But what we are learning here, in photosynthesis, electrons were actually going from high reduction potential to low reduction potential, and the system has to design a grand scheme of Z scheme, and here it is not happening like that. The electron transport is straight. It is continuously going from NADH to 1 to 3 to 4 and ultimately to oxygen in increasing order of reduction potential. So we have learned that it is the tendency of accepting the electrons. We have learned that electrons are flowing from low reduction potential to high reduction potential. We have learned that this small concept is having very high impact on designing of bigger designs or bigger pathways like Z schemes and electron transport system. So it was very essential to understand the logic of reduction potential. I hope you have learned it, and if you are having any doubt, you can always ask us in the comment section, or you can always write to us. So now I am going to summarize our life based on reduction potential. The life starts from water, which is giving us oxygen and giving us electrons. These electrons are given to NADP plus, which is converting into NADPH in light reaction. NADPH is converting back into NADP plus, and the released electrons are given to CO2, which is making glucose. Animals, plants, different type of cells are utilizing this glucose, again generating CO2, releasing the electrons. These electrons are given to NAD plus, which is converting into NADH. It is recycled into electron transport system. NADH is giving us electron. Those electrons are given to half O2, which is again converting into H2O. So the life starts from water, take a midpoint at glucose, and we call it as photosynthesis. From glucose, the remaining life cycle starts, ends at CO2 and ends at water. We call it as respiration. So if I summarize the life on this planet, then it starts from water and it ends at water, and in between what we are having, flow of electron through different kind of pathways, different kind of designs, different kind of biochemical processes.